Hi again Year 9 and welcome back to your final half term of science. Hopefully you've all had a nice break over the half term holiday and now you're ready to come back and complete some science work. So what I want you to do is make sure that your phone is turned off or in a different room, and then you don't have any other tabs open on your computer except this video, and then you might have the electronic booklet open as well. So you need a copy of the booklet, which is called Genes, DNA and the Cell Cycle, and you can either have this printed or electronically, or alternatively you could just work on paper with the booklet open electronically and then send us a photo, your science teacher, a photo of the work you've completed on the paper at the end of the week. I want you to try every single question, pausing the video when you do answer the questions. I'll tell you when to pause the video, and then remember you can always rewind the video and go over a certain section again if you're struggling on that section. I also want to remind you that you can go onto BBC Bitesize, so if you just Google BBC Bitesize Cell Cycle, the relevant pages will come up. And then finally, don't give up. You've done absolutely fantastic so far, Year 9. This is your final term. Keep going, keep working as hard as you have been. Just finally, before we move on to the next slide and we get started on the lesson, you're also going to need a calculator for this lesson, and then if you are working on paper you're going to need a pen, a pencil and a ruler. Okay, let's get started then, guys. So the first part of your booklet is the Do Now task. It has a diagram. You've studied DNA in Key Stage 3, you've actually studied DNA and genetics a little bit in Year 9, so I want you to have a go at labelling the diagram and then answering the questions underneath the diagram. The answers are on the next slide, but I want you to have a go first. You can type your labels into the boxes on the documents, or you can just do a rough sketch on a piece of paper and complete the labels on there. So give yourself about 3 or 4 minutes to complete this, and pause the video now and then restart it once you've had a go. Okay, let's see how you've done then. So if you're working on a piece of paper, a different colour pen would be useful now to mark your answers, or you might be marking them on the Word document. So the label A, we can see is pointing to the nucleus. You should have got nucleus there. So the whole structure that A is pointing to is the cell, but then in the middle of the cell is the nucleus. And remember, that is the part of the cell which holds the DNA. We then have B, which is that X structure. B is a chromosome. So remember, we have pairs of chromosomes as humans. We have 23 pairs of chromosomes, and all these chromosomes look a little bit like an X. So you will see them in diagrams drawn like an X. And then C, remember, a section of DNA is called a gene. So what's happened is it's unravelled that chromosome to get to the DNA, and a section of DNA is called a gene, and C is pointing to a section. And then the final one, if we go down to the smallest structure in this diagram, D, it is the DNA. So DNA is the smallest structure of genetic information. So chromosomes are found in the nucleus. They are made from huge strands of DNA. A small section of DNA is a gene. So you might want to jot down the definition of a gene. It's a section of DNA. So chromosomes, for your question, chromosomes are in pairs in body cells because in the original fertilised cell, half of them came from mum and half came from dad. So we inherit 23 chromosomes from mum and 23 chromosomes from dad. That's why they are in pairs. So what we're going to be looking at this session then. So this is still in the cells topic. We're coming to near the end of the cells topic now, but we still need to learn about the cell cycle and cell division. So You're going to be identifying DNA, genes and chromosomes on a diagram, which we've already covered in the Do Now task, so well done, you can tick that first one off. The next outcome is to describe the main stages in the cell cycle. So when we talked about cell cycle, we just mean the life cycle of the cell. And then the last one, we're going to use information provided to calculate time spent in different phases or stages. So on this slide you can see many different species. You can also see the number of chromosomes that each species has. I want you to notice how in every species the chromosome number is an even number. And this comes back to what we discussed in the Do Now task. This is because half of the chromosomes come from mum and half of the chromosomes come from dad. So it has to be an even number. You can also see in this diagram that there is no real connection between the number of chromosomes and the size or complexity of an organism. It seems almost random. You can see in the bottom right hand corner there's a plant which has 48 chromosomes in comparison to a cat, which is a much more complex organism than a plant, and cats only have 38 chromosomes in their nucleus. What do we mean then by the cell cycle? So cell cycle is just the different stages of cell division. So all animals and plants, all of us, have all started life as a single fertilised egg cell. But an organism, like us, is made up of billions of cells. So to get from that one single cell, which was the fertilised egg, to billions of cells is going to require cell division. Lots and lots of cell division. Cells are going to have to divide billions of times. All organisms also need to regularly replace damaged or worn out cells. So we will be carrying out cell division as we sit here listening to this lesson. Cell division will be occurring in our bodies. So we're going to go through in the next few slides what happens during cell division and the different stages of cell division. Okay, so on this slide I want you to have a look at that diagram on the left, and inside the diagram you can see those different colour blobs, or chromosomes is what they are. We can see that in the second cell down from the top we can see that they look like the X's that I mentioned earlier, but in the top cell they don't look like the X's, and this is because they are split apart. I told you that they pair up, so in the top they are single chromosomes, but then in the cell second one down they are paired chromosomes, and when they are paired they look like them X structures. So I want you to look at the diagram. Don't write anything down yet, just look at the diagram and think of a sentence to describe what is happening at each stage starting at the top. So we can see several different stages there as we go down. I have some words to help you. So chromosomes, copied, lined up, membrane, divide and new cells. I want you to think about how many chromosomes we have in each cell, which will help you to see whether we have doubled or halved the chromosomes. How do the new cells compare with the one at the start? I've also highlighted this word at the bottom, so mitosis. For this slide, I'm just introducing you to this word. It's a scientific word for cell division. So mitosis just means cell division. So have a look at the diagram on the left and try to come up with a sentence to describe what's going on at each stage using the words to help. Okay then, so don't worry if you struggled with this task. We're going to look at each stage in a lot more detail over the next few slides. But let's have a look briefly at what happens during cell division then. So in the first stage we have 4 chromosomes. So this is our first initial cell. And we want that cell to divide, but when it divides we want 2 identical copies of this cell. So how are we going to do that? We're going to need double the amount of chromosomes, because when they divide, half of them will go into one cell and half will go into the other. So in the first cell we can see we have 4 chromosomes. In the first stage of cell division, each of the chromosomes are copied to get 8 chromosomes which pair up. So we can see we now have double the amount of chromosomes. That's what we need. Then the next stage, the chromosomes line up in the middle of the cell in their pairs, and then they're in this perfect line in the middle of the cell. The chromosome pairs then start to move apart and divide, so half of the chromosomes will go to the left and half will go to the right because they're going to become part of new cells. Then we need the cell membrane to divide, so the cell membrane starts to split in half. Half of it going to the left, half of it going to the right. And then finally, we can see in the last stage, we have 2 new cells that have been created that have the same number of chromosomes as the first cell. So each of the new cells are identical copies of that first initial cell. Each of them has 4 chromosomes in, and that's what we needed. So as I mentioned before, mitosis is the fancy scientific word that describes cell division, and it's the type of cell division that happens during growth and repair. So we're going to have a look at what happens during mitosis, during cell division, in more detail, and we'll come back and describe those diagrams in more detail later on. So during cell division, the entire cell contents needs to be copied, and then the cell divides in 2. This means that ribosomes, mitochondria, all have to be copied, because all of the new cells are also going to need ribosomes and mitochondria. So we're going to need 2 of everything, so that the 2 cells that we create at the end are identical. So all of these stages in the cell cycle have special names, and we can see in that pie chart at the bottom, in the bottom left, how long the cell spends in each stage in the cycle. So we can see that interphase is where the cell spends most of its time. So in interphase, which is the first stage, the cell starts to grow, and the number of subcellular structures, which is just organelles, such as mitochondria and ribosomes, increases. So we have to double all of them, so we can be ready to divide the cell. The DNA also replicates, just means copies, doubles itself, to form 2 copies of each chromosome. We also check the DNA for errors and make any repairs to the DNA, because if the DNA has some errors into it, it could code for something dangerous. It could cause the cell to die, which we don't want that to happen. Remember before I told you that mitosis is a fancy name for cell division, and mitosis is one of the main stages in the cell cycle. So in mitosis, the chromosomes move apart, so the chromosomes have lined up in a perfect line in the middle of the cell, and they move apart, and then the nucleus, where all the chromosomes are, divides. So we get that first initial stage of cell division. the genetic material splits in two. The final stage, and notice from the pie chart we have interphase as the longest stage, and then mitosis and cytokinesis are more or less identical for time length. So in cytokinesis, this is when the cytoplasm, so remember mitosis is when the nucleus divides, but in cytokinesis we also need the cytoplasm to divide. So the cytoplasm divides into two, and the new cell membrane separates off to give two new identical cells. This is the final stage, and at the end of this stage we have our two new identical cells, which are a copy of the first initial cell. Okay, so I told you we were going to come back to this diagram. I want you to try and not rewind the video for this task, because we went through some similar answers earlier. In your booklet you can see you've got the diagram. I want you to try and order the steps underneath the diagram into the correct order. And if you're working on your computer, you can drag and drop the boxes with the sentences in to match the right diagrams. Alternatively, you could just sketch the diagram yourself, even just drawing circles is fine, and then you could write out the different stages. So you need to decide which order they go in, and then I also want you to drag the names of the stages to the right place. So which part is showing interphase, which part is showing mitosis, and which part is showing cytokinesis. We didn't actually do the interphase, mitosis and cytokinesis on the last diagram, so this is an extra challenge for you guys now. So you're sorting all of the stages into the correct order, either sketching, writing them out, or you can drag and drop the box if you're working on your computer. Make sure you read all of the sentences properly, and I'd give yourself about 5 minutes to complete this task. So pausing the video now, no cheating, no rewinding the video to look back, having a go yourself, and then we'll go through the answers in about 5 minutes time. So let's have a look at how you did then. So the first stage was already done for you. So the original cell has 2 pairs of chromosomes. 2 pairs means that we've got 4 chromosomes altogether. Then you should have said that the chromosomes are all copied as the next stage. Different colour pen would be useful here. After the chromosomes are all copied, the next stage down, the chromosomes line up in the centre of the cell. After that, the chromosomes are pulled apart as the cell begins to divide. Then the nucleus divides, and then finally the cytoplasm divides and new cell membranes form. So check your answers and correct any that need it. I also asked you to label which stage was interphase, which stage was mitosis, and which stage was cytokinesis. So interphase, remember, where the cell spends most of its time when all the chromosomes and all the organelles are doubling, are being copied. So the first 2 cells are showing interphase happening because the chromosomes, the DNA, is doubling. We then have mitosis, which will be the next 3 cells down. Remember, in mitosis, that is where the DNA starts to divide. So the nucleus is starting to divide. So that involves the pairs lining up and the DNA, the chromosomes, starting to move to opposite sides of the cell. And then the final stage, cytokinesis, which involves the cytoplasm dividing, is the last two cells. So that final stage where the cytoplasm divides and we have our new two identical cells. We touched on this before as we were going through the different stages, but this pie chart demonstrates to us the relative time spent in the stages. So we can see most of the time is spent in interphase, that blue part of the pie chart, and then we have much less time spent in mitosis, where the nucleus is dividing, and then we have about the same time in cytokinesis. So you will have looked at some cells down the microscope before when you've been in school. And when we look at cells using the microscope, because cell division is constantly happening for growth and repair, it's happening right now in our bodies, if we took some of your cells and looked at them down a microscope, we'll be able to see some that are actively dividing. And I've circled the ones that are actively dividing in red. And we can see that they are actively dividing because the nucleus isn't just a blob. The nucleus has started to separate all into those separate chromosomes, and the chromosomes are moving apart in some of them. So in some of them the chromosomes are just lining up, and in some they're actually moving apart. There are a few more cells that are dividing on top of the ones that I've circled. Can you find those cells? Anywhere that you can see the chromosomes lined up in the centre or being pulled apart from the centre is an actively dividing cell. And I just think it's absolutely amazing how we can see this using a microscope. For the purpose of this slide, what I want you to do is think about mitosis as two separate stages. Because remember, when we labelled that diagram, mitosis was labelled as two of the separate stages. So instead of it just being interphase is stage 1, mitosis is stage 2, and cytokinesis is stage 3, we're going to have interphase as stage 1, mitosis as stage 2 and stage 3, and then cytokinesis is stage 4. So we can use the number of dividing cells to estimate the length of each stage of the cycle. So we can estimate how long a cell spends in interphase, how long is the mitosis stage, and then how long is the cytokinesis stage. So if on the previous slide we'd have counted up the number of cells that were not dividing and then the number that were in each of these different phases of division, we'd get a table that looks like something like this on the slide. So we can see we have 20 non-dividing cells, we have 5 cells in stage 1, interphase, 1 cell in stage 2, 8 cells in stage 3, and 9 cells in stage 4. And this gives us a total, if we add them all up, of 43 cells. So if the whole cycle lasts 24 hours, then the length of each stage can be estimated by expressing the number of cells in each stage as a fraction of the total number of cells. So for example, we know that in stage 3 there are 8 cells, and this is 8 cells out of a total of 43 cells. So if we work this out as a fraction, so 8 over 43, and then times it by the amount of time for the whole stage, which is 24 hours, we can work out how many hours a cell will spend in stage 3. So if we do 8 divided by 43 times by 24, then we should get an answer of 4.47 hours. And if we times this by 60, we can get it in minutes, which would give us a time of 268 minutes. Don't worry if you're a bit confused by this, because in your booklet you have a step-by-step table which helps you to work this out. So now I want you to have a go at this question here. This question is in page 2 of your booklet underneath the diagram that we labelled earlier. So I want you to stop the video while you have a go at this question. I'm going to go through the answers on the next slide, but I want you to try it yourself first. You will need a calculator to do this. So for this question, it tells us that the whole cycle lasts 13 hours, not 24. Just remember, we're going to times it by 13, not 24. I want you to estimate the length of stage 1. Express your answer in minutes to 2 significant figures. So remember, at the end you're going to have to times by 60 to get it into minutes instead of hours. Use that table in your booklet which goes through the steps in order to work this out. So it tells you what to do on the left hand side of the table, and then you need to complete your answer for each stage in the right hand side of the table. And then we're going to go through the answers on the next slide. So pause the video now, have a go at this question, and then we'll go through the answers. Okay, so hopefully you found that question okay. The first step in the table was to tell me how many cells are in stage 1. So we can see there are 5 cells in stage 1 out of a total of 43. The next step was to divide the number of cells in stage 1 by the total number of cells. We can see at the end it tells us the total number of cells is 43. So what we had to do is do 5 divided by 43. Once we've worked that out, it asks us to multiply the answer to step 2 by the length of the cycle. So we would do 5 divided by 43 times by 13 hours. And then the length that we get is 1.5116 hours. We want this in minutes though, remember. So the next step was to multiply the answer by 60 to convert into minutes. So the answer in minutes is 90.697 etc. minutes. And then remember, it asked us to work out our answer in two significant figures. So the answer would be 91 minutes. Okay, so now is a chance for you to check your learning during this lesson. So I want you to think through the answers for each of these questions. You do not need to write the answers down, but definitely have a think about them. Some of these questions are going to appear in the Do Now next lesson. So stop the video now, have a think about the answers to these questions. So the first question we first looked at right at the start of this lesson: why are chromosomes in pairs? They have to be in pairs, they have to be even, because one chromosome of each is inherited from mum and one chromosome is inherited from dad. Name 2 reasons organisms need to carry out cell division. They need it for reason 1 is growth, reason 2 is repair. Number 3, what type of cell division is used for growth? It's that fancy word that we looked at, it is mitosis. There's another type of cell division, but you won't look at that until year 11. Question number 4, which phase in the cell cycle is the longest? It has to be interphase. Interphase is significantly longer than mitosis or cytokinesis. What happens during interphase then? The cell grows and the contents such as the organelles and the DNA is copied. Question 6. What happens during mitosis? So the chromosomes are pulled apart, they line up, they are pulled apart to opposite sides of the cell and the nucleus divides. Question 7. Which part of the cell cycle involves the division of the cytoplasm and the cell membrane? It is cytokinesis. So remember, some of these are going to appear in the Do Now task next lesson. So the final part of this lesson is for you guys to have a go at the exam questions. All these exam questions are taken from past GCSE papers. So it is really important that you're having a go at these questions in preparation for coming back to school. As well as the exam questions on the continuity of learning section science, there's also the mark scheme for these exam questions. So give yourself about 15 minutes to complete the exam questions and then mark your answers. That's all the content covered for this lesson. As well, in your booklet you also have the fluency test, which should be giving yourself about 10 to 15 minutes to do. Remember, on the fluency test you have your hypothesis, your scientific prediction at the top, and then you have several questions relating to that hypothesis to answer underneath. The mark scheme to the fluency test is also uploaded in the science section. So you should now do the exam questions and the fluency test. You might want to leave the fluency test for another day, but you do need to complete it, and you do need to send a photo to prove, or a screenshot or the Word document to prove that you've completed these, to your science teacher through their email. All emails can be found on the school website. And then remember, you've also got that compulsory Educate quiz to complete, which needs completing by 8 AM on Monday morning. So once you've completed all of that, I just want to say a massive well done again, guys. That's the end of the content for this lesson. Keep working hard. Well done.